The overall goal of this procedure is to more easily visualize fluorescent neurons within thick brain slices. This is accomplished by first embedding the brain in an agarose plug holder. The second step of the procedure is to section the brain into thick slices on the compressed tome. The third step of the procedure is to optically clear the brain slices in a glycerol gradient. The final step of the procedure is to mount the cleared brain slices onto slides for fluorescent imaging. Ultimately, results can be obtained that show high-resolution fluorescent protein reporter expression in neurons within thick brain slices through epifluorescent or confocal microscopy.
The main advantage of this technique over existing methods like thin-section serial fluorescent microscopy or multiphoton imaging is that it is much more rapid, less labor-intensive, and does not require expensive or sophisticated microscopy technique.
To begin, retrieve the mouse brain tissue that has undergone in situ fixation by cardiac perfusion and has been post-fixed for 1 to 2 hours at 4 degrees Celsius. Next, make a 2% high-strength low-gel point agarose solution in PBS and heat in the microwave until dissolved. Transfer the molten agarose to a test tube and place in a 40 degrees Celsius heat block to equilibrate the temperature. While the agarose equilibrates, glue the fixed brain tissue onto the plunger of the specimen syringe with cyanoacrylate glue. Submerge the mounted tissue in a saturated sucrose PBS solution to coat the brain surface, and then draw down the plunger into the plunger housing. Pipette the warm agarose into the plunger, completely covering the brain tissue while making sure not to introduce any air bubbles. Finally, clamp the specimen syringe with a cold chilling block for about 1 minute to set the agarose. Assemble the specimen syringe